Okay, thanks, David. Good morning, everyone. I'm Greg Daniel. I'm Deputy Director at the Duke-Margolis Center for Health Policy. And as was opened with Greg and David, we're now turning to our last essential question for exploring the potential decision aids. The specific question is, how can bias and observational comparisons be assessed and minimized? And the more broader question, and we're going to discuss that on our panel, but also the more broader question of when do we think that observational studies can be credible enough to support regulatory decisions? This is something that everyone is grappling with, and it's an area where there seems to be a lot of discomfort as well. On data quality and relevancy, which we discussed a lot yesterday, we generally have a good understanding of what constitutes good quality data and highly relevant data. As you heard a lot of yesterday, the Duke-Margolis Center is doing some work around this question of real-world data quality and relevancy across a range of data sources. But we still need to think, and I think a lot of remaining questions from yesterday was, when is good enough? You have various levels of data quality, and do we have to sacrifice everything to get the perfect when we may not need that? And where do you sort of draw the line, particularly as it relates to which regulatory question is being asked and what the clinical context is? We're also seriously grappling with the question of credibility of observational designs. And again, we heard yesterday, grounding everyone in well-established epidemiologic methods and approaches makes sense. ISPOR, ISPI, and a lot of groups have been looking at this for decades. How do we do really good observational designs? We know how to do that, and I think this decision aid sort of walks us through systematically is how to make sure that we're making the right decisions about designing our observational designs and we're using them in the right way. However, what we really need to get to is how do we know that the observational design that we're using or the series of observational studies are good enough given the regulatory question and given the prior knowledge of the drug that we have, presumably from Cures and FDARA, where FDA most likely might start out utilizing real-world evidence beyond how it already does, is in the context of a drug that's already on the market and we're looking at potential new indication or labeling changes. So we know something about that drug already, and most likely from randomized controlled trials. So a good question is, given what we already know about the drug, our prior, where are the gaps in evidence that we're trying to fill with observational designs, and how can observational designs best fill that evidence? So it brings us to questions like, when can observational studies be relied upon given the regulatory question and what's already known about the drug? How, in those contexts, can we make the study more credible, and how do they fit into that overall body of evidence that will be generated on that drug as we learn while patients are, once the drug is marketed, and we start learning and accruing evidence on that particular product? To this last point, a key component of the work of the Duke-Margolis Center is exploring how best to anchor discussions on the acceptability of observational designs and approaches to the well-established substantial evidence threshold that FDA uses when making decisions about medical product labeling, new indications for already known approved medical products. So within the context of the totality of evidence, what is already known about the drug, how can observational studies add to our accepted understanding of the safety and effectiveness of the product? And how can a submission package that's unlikely to be a single observational study, but most likely a range of studies that include observational designs, perhaps multiple observational designs in different populations, how can that totality of evidence be weighed when considering the particular clinical question and regulatory decision? So the decision aid for reducing bias in observational designs that David just walked through is a really good start to think on an individual study case. How do we make sure that we're making the right decisions and that we're generating an observational study that's highly reliable and potentially acceptable? So the question is, how does that map to substantial evidence, and how do we consider what's already known about the drug when we're designing those studies? So with that, I'll go ahead and introduce our panel and ask that they that they join us. So Hector Izurieta is epidemiologist in the Office of Biostatistics and Epidemiology at CBER, FDA. Jessica Franklin is assistant professor of medicine at Harvard Medical School. You can start coming up. Nicole Gromley is team lead, Division of Hematologic Products, Office of Hematology and Oncology Products in CDER, FDA. Javier Jimenez, vice president and global head of real-world evidence and clinical outcomes at Sanofi. Hang Lee, mathematical statistician, Center for Devices and Radiologic Health at FDA. And then Mark Vanderlaan, professor of biostatistics and statistics at University of California, Berkeley. So I'll go ahead and turn things over to Hector for his presentation. Thank you.